Okay, take home point from the earlier set of slides we just completed. There is a workflow that allows you to pre-process fMRI to get ready for the main show, which is actually creating what we call the functional network or the functional connectome. And here I'm going to actually motivate why that's important, how that's done briefly, and then go back to why it was important to actually register to physical locations in the brain. It will become clearer. So fMRI is an old technology. Roughly, I think about, again, roughly 20 years from the 1990s onwards. And the processing framework that was built up actually came from one particular worldview. And that worldview is that a specific location in the brain is assigned or has responsibility for one function. So one function is due to one location in the brain. So the goal of mapping brain activity under this old paradigm was, I give you a task and I try to figure out which part of the brain lights up, and I say that part of the brain is responsible for that task. Okay, so this is kind of a region parcel based method. And the way the studies tend to be designed was, I interleave the task with a do-nothing resting state. So I'm taking your fMRI measurement continuously, but maybe the first 30 seconds I give you the task. The task could be selecting something, or recalling a word, or making some movement. And I'm recording your blood flow changes, and then for the next 30 seconds after that, you don't do anything, you just take a break. And then the next 30 seconds after that, you do the task again. And the next 30 seconds after that, you don't do anything. So you end up in a single fMRI acquisition. You have the blood flow activity during the task and when the brain is not doing anything. And you make a statistical comparison, looking at what regions were activated during the task and what regions were not activated during the rest. That's the region that people say would then be associated with that task. So again, that concept is one brain region, one task. It's hypothesis driven, meaning that you have to have a hypothesis that doing this task will activate new brain regions, that brain regions are responsible for something. And the analysis comes under the generalized linear model. I'm not going to go through this because this is not the network view of things. So this is just a visualization of what that process could look like. Okay, someone is doing you start first with taking you're constantly acquiring images, okay, of someone not doing anything, and that's this control period, and maybe one particular voxel will have this activity, and you will be able to get a map of activity of the entire 3D brain. And then when you are actually performing the task, that activity at some voxels will be greatly increased or significantly increased. You have a map of those increased area, and you just do a kind of a subtraction and you run some statistics on that. That's kind of the key idea. You're just comparing between what's the activity during the task, what's the activity when you're not doing anything. Because when you're not doing anything, the brain still has some activity that's regarded as the noise, and only the significant changes during the activity is considered to be the regions that really significantly activate. The caveats of this view, we've observed from all these studies which have been going on for 20 years, most brain regions are activated for more than one task, right? So if you have a memory task, you have a cognition task, you're walking, you actually see some of the brain regions activating for more than one task. So how do you make sense of that? Are you saying that this particular brain region is doing all of that? Sure. Right? The far end of that, you say your whole brain is doing everything. Sure. Right? So that's less informative. And something that actually came up was people in the early 2000s looked at the fMRI of a person's brain while they were resting, and they found that there was significant activity during this rest period. So your brain is actually very, very active. It's a lot of blood flow changes, even if you're not doing anything. That's why it's called a resting state fMRI. And what's more interesting is using some computational techniques, they will be able to separate certain regions of the brain that tend to activate together. Okay. So now I'm looking at a resting brain, and I'm finding that there are certain regions of the brain that they like to activate together. If one increases, that region will tend to increase. If it decreases in blood flow, that region tends to decrease. And so these regions of the brain that tend to be in synchrony, they increase and decrease kind of at the same time, these people start to think about them as maybe a network, because they probably are working together because they are kind of activating together. And what's more interesting is when people have done the analysis on these activity patterns, they're able to extract out several of these networks that are occurring at the same time. Right? So these are what we know as the resting state networks. And this is from a paper that shows a sampling of some of these networks. And what's interesting is these same regions that activate together, the networks, seem to be conserved across a whole lot of people. So I'll repeat what I said. The caveats people start to see is certain brain regions seem to activate in a kind of synchrony. It's not a perfect synchrony. You can actually put a value to it, like how co-activated they are, how correlated they are versus totally uncorrelated. On the far end, they are totally uncorrelated, meaning that whatever this brain region, how it changes, doesn't actually affect how this one changes. But what we've seen is that there are some parcels or regions of the brain that tend to work together. And when you're looking at the total activity of the brain going on, you can actually separate out different sub-networks that work together. So for instance, these yellow regions here would tend to fire together, but they will be firing at the same time as these regions, but these two regions will be firing, but they are not related to each other. It's just activity overlaid on activity. And all these networks can be quite reliably extracted from different people. And equally as interesting, you see these same concepts applied while someone is doing particular tasks. It's still the concept of the network, the functional network. And by functional network, I just mean a group of brain regions that tend to have some kind of synchrony, some kind of correlation, they activate together. So this was seen when you are doing a task, and it was also seen when you are not doing a task. But they're not exactly the same regions of the brain when you're not doing a task versus doing a task. There are a lot of differences. Okay. So because of that, this has motivated a new view of the brain, that your brain actually functions as a network of collaborating cells. And it is recognized that the same cells can actually be collaborating with different networks at the same time, because the networks are overlaid. And this gives rise to the concept of a functional network. So what is a functional network? So we start by saying, let's take a look at the whole brain, and we split it up into groups, or I like to call them parcels. Some people call them regions, segments. It's split up to groups, and within each group, the activity is the same. The blood flow changes in sort of the same way. Now you're looking at all these parcels, okay, and you're trying to figure out which set of parcels have synchronized activity. If they have synchronized activity, you can start, so you can represent each of these parcels as a circle, and that's called a node in graph speak, because it represents a group of cells or a physical location in your brain. Now, if two parcels kind of have a correlation, that means they are synchronized in their activity, you can draw a line between them. And you can put a value on that line, and that value could indicate the degree of synchrony between the parcels. And you can do this for combination of every parcel the brain, and you end up getting a graph that looks like this. So let me try to rephrase and repeat this. So you start with the functional images, a whole stack of time series brains, okay, and you get a time series from each voxel, okay, and at the same time you need to be able to break up the brain into parcels or nodes. What makes sense to group together? Which voxels make sense to group together? There are many different ways to do that. Okay. And then you will look at a group of voxels in a parcel, and you will compare their average activity to the average activity of the second parcel and to each and every other parcel to see how synchronous they are. And by synchrony, you just do like the Pearson correlation. And that Pearson correlation tells you the average synchrony between each pair of parcels. And doing this, you can actually reconstruct a network, basically combining of individual nodes, which are your brain parcels, and lines connecting those nodes, where the presence of the lines indicates synchrony between those nodes. And each line, called an edge, has a value associated with it, or maybe even several values, but for now you think about it as one value, and that value is just a measure of how synchronized they are. And you can represent this network also as a two-dimensional matrix, right? So if you were to label each location, each column and each row as the name of one brain parcel, so brain parcel A, brain parcel B, you find the point where they intersect. The value in that little box, in that matrix, is the value of the synchrony between those two parcels. And so I can construct a 2D matrix where I compare every parcel against every other parcel, and I put down their value of synchrony there. That's another way of representing a functional network. So this is a way to visualize the functional network, but that network can also be represented as this. And sometimes people say, I'm only going to consider a functional connection if a synchrony is above a certain value. And that leads you to a binary connection diagram where instead of having a number between 0 to 1 that gives you a measure of that connectivity, you say, if it's above 0.64, then there's a connection, otherwise there's not. So there's a connection means there's a red dot, if there's no connection then it's a blue dot. Okay. So there are kind of two approaches to analyze functional networks. One is you are using a continuous value. So I've just walked you through the first and easiest method to construct a functional network. That is, you have one reference parcel or one reference node, and you compare every other node against it, and you get one functional connectivity matrix. It's just one way, and then your reference point is to one particular brain region. There is another way of extracting the functional connectome. That is a statistical method called independent component analysis. You can actually just take the brain either as a group or as a single brain. If you run ICA on the time series of the brain, it will extract out the locations that are activating dependently on each other. So one way to think about it is the ICA, the independent component analysis, extracts out networks or parcels. One network or parcel is going to be independent in time from the second network of parcels, going to be independent in time from the third network of parcels. So that's what the ICA does. And why is independence a reasonable property? Is because, well, if the network's doing if the two different networks are doing a two different things, we don't expect there to be any kind of relationship between the activity of one versus the other. Because remember, how do we define the existence of a functional network? They co-fire, they co-activate together with some degree of synchrony. So there are two general approaches, and again, ICA is not the only technique you will be able to extract from that. There are variants of ICA and all that to be able to extract. People have been working on a lot of this. Okay? But these are the kind of like the two big picture general approaches. And recently there's a movement to actually connect these functional networks to more to some models of the brain that are more probabilistic in nature, causal models, dynamic causal models. They are probabilistic models. You can take a look at Professor Visten's paper on that, trying to actually link the functional connectivity matrix and matrices to actual statistical models of dependency between this region and this region, because he's trying to reason out, he and others are trying to reason about what brain regions cause which other brain regions to fire, what is the causal relationship between an activity in this network versus another network or inside the network. So these are the interesting directions where functional connectivity is doing. So take home point from this, you have a high level view of how the functional connectivity matrix is created through general ways, and you know that people are working on many other different ways to develop those matrices. People are also looking at trying to link those matrices to more informative models of the brain, and you will be working with these matrices tomorrow. Now, yes?
This network, like this matrix, if we look at the sequence of the time series, the signal, can we move a window toward the signal with an overlap, actually half-percent overlapping in order to move the signal into the frequency domain?
Okay, Amin is asking an interesting question and many interesting points. I've just told you about synchrony, but synchronization happens within a window. And the really, really interesting thing about these functional networks is they evolve over time. And so we actually should be thinking about the dynamics of a functional network. On what time scale. So that sliding window, the width of that window and how you slide that could capture the evolution of the synchrony. So you have a network, okay, a network which is you define as functional synchrony, but that synchronization will evolve over time. It changes over time. And I think people are looking at the change in the functional connectivity as a kind of representation of mental state, which is really interesting. Right? There's a work, the one from this paper, Scherer's paper. What they did was they obtained the functional networks of the brain in 4 different tasks, okay, and then they create a machine learning classifier so that you can tell which of the tasks it's working on, and then they fed in data from a 5th task that it's never seen. And that classifier could actually say, this is a different task, I've never seen this, and it could reliably predict the 4 different tasks to which it was trained. I think the very coarse, not very strong conclusion that I'm making is people are starting to see some evidence that the functional network could represent different states of the brain, different mental states of the brain. And there have been work also looking at how the networks can evolve from one state to another state, and that evolution of the networks, the properties of the networks have been linked to intelligence, have been linked to disease and degeneration. We'll talk about that a little bit more, not too much longer, but a little bit more. And that's why network analysis is so important to me. It's so interesting, because there's this big field of why it's important. So functional connectivity networks, again, it could be task-based networks, or it could be resting states. The networks are dynamic. The network actually changes over time. And that's why the data set that I'm giving you, the Raspodrek's MyConnector data set, is from the same person, imaged throughout 1 year, every week, and for 20 minutes. And they have done some analysis. They can actually see the shift in functional connectivity in a person, and their work actually showed that there are some elements of the functional network that is conserved across individuals, and some parts of the functional network that seem to be unique identifier of each individual brain. So this field is really big, wide, growing very rapidly, lots of new findings to be extracted. So you more or less know how to interpret this kind of graph, because we talked about it, that each of these nodes, these balls, the node, represents a brain parcel, and the line connecting brain parcels indicates the presence of a functional connection, which just simply means the two nodes that have a line between them have some kind of synchronous activity, okay, correlated activity. We can actually represent these connections in a circular diagram where this again could be each of the brain regions, and you can actually draw a line, you can change the color of the line, you can change the thickness of the line, just to give you a visual representation of the network. Yeah, but this actually isn't a functional network. Today I'm actually just focusing on the functional this afternoon, because tomorrow you'll be working on functional networks. But the same analysis can be also run on structural networks. And you also now understand from this morning how structural networks are constructed. Same graph properties. The node is actually a brain parcel. The line connecting each node is actually the existence of a track or not. And the value of that line could actually represent the thickness, how strong, how many tracks are actually present there. It could represent things like the property of diffusivity, like how leaky are the neurons along that line, or how many tracks are actually there, how dense are the fibers, a lot of different parameters could be represented. So it's a rich space for exploration. So I'm not doing justice to functional networks, but I'm just going to give you one key reason why I think functional networks are important. There are of course others in the performance domain, mental states, looking at brain ability, transition between states. But what really interests me is people are finding evidence that they see changes in the functional and structural networks in disease. And basically they are looking at cohorts that have various kinds of neurological disease, psychiatric disease, degenerative diseases, and they're seeing key differences in functional networks, as well as structural networks. And so where are these differences? They lie in the dimension of the number of connections, number of links. So maybe in a disease group versus a non-disease group, the disease group may have more connections between parcels, or new connections could exist between parcels that did not exist in the normal cohort. Okay. The strength of the connections could be changed. Maybe in the disease group, it could be a weaker connection. In the non-disease group, it could be a stronger connection. How the connections evolve in their interactions with each other. So this is kind of a high order idea that. So I talked to you about a functional network, but then you also understand that there are several different functional networks all operating at the same time. So I'm not sure if people have constructively looked at the interaction between how functional networks work at the same time. So that could be another interesting area to look at as well. And how the functional networks change over time. And this is not something that has been widely studied as well. But it is known for a fact that functional networks will evolve over time, and they evolve as functional brain states, and people have done theoretical studies showing that the reachability of the states, like your brain is most intelligent when it can reach a certain number of states most easily in the fewest steps, something along those lines. So there are mathematical formulations of that. But the key point really is that differences in the functional and structural networks seem to be consistent and stably present between disease groups and non-disease groups for what seems to be a broad range of neurological, psychiatric, and neurodegenerative diseases. And one example, maybe in the neurodegeneration, sometimes you see that the number of links in the network actually grows, and the physical interpretation that people are giving is that maybe there's a local failure, and so the brain is trying to compensate by increasing connections, by recruiting more cells. Or on the other hand, if you actually see the number of connections reducing, it could actually be a degeneration and damage. So these are some of the interpretations that grow. So I just show you these slides to actually let you see that there's this range of diseases here from this paper, where they've looked at different functional networks. Here they actually represented it as just blobs on 2D MRI images, and whether there's an increase or a decrease in the number of connections. And you can see for some of these diseases there are like really large, significant changes. And I think this value is probably one of the statistical tests, maybe 1 SD or 2 SD. But the key point here is not just for one disease, they've actually looked at a bunch of diseases, and they've actually seen that some diseases, clearly you can see a shift and a change in the functional networks. And here's another long list. You can trace it in the papers. And equally interesting, there was a meta-analysis done looking at 392 studies of 26 brain disorders, and they only looked at the structural network. And they found that the common theme in most of these diseases was a damage or a change in certain particular brain parcels. And interestingly, those brain parcels are what we call hubs. They are hubs because they are locations where they are of high connectivity. Lots of other brain parcels are connecting to those regions. So this paper, this meta-analysis seems to suggest that damage to brain regions that are highly connected to other brain regions seems to be a possible underlying factor of many of these neurological diseases. So if you see a damage to a hub region, a hub parcel in the brain, chances are it could actually lead to one of these diseases. So this is some of the common observations that people are starting to obtain. And another interesting point, they are actually looking at the correlation of functional networks with changes in genetics, the distribution of genes, as well as the distribution of various kinds of tau proteins and some of these residual proteins that are associated with degeneration. And they are now, and they see an overlap in the locations of aggregation. And so people are actually chasing down, and I think there's preliminary evidence that the functional network appears to be kind of like a conduit, a pathway for which some of these proteins and degenerative processes can spread. This is not fact, it is just some preliminary evidence pointing towards this possible interpretation. But that makes the idea of a functional network important, right? Because a functional network is actually a representation of activity in the brain. It seems to change with disease and degeneration, and it seems to be linked or overlaid with the spread of some of these physical pathologies that we see that are associated with neurodegenerative diseases. So there might be a story that links everything together. So again, changes in the functional networks during neurodegeneration. Okay, so this is a big picture motivation of how you can actually assemble a functional network, and the representation that you'll be dealing with in the computer is a two-dimensional matrix. Of course, to get to that particular compute, you will again be using a workflow from Docker, CPAC. Okay, so not to worry too much about the mode of calculation, because based on the diverse background, I'm not presuming that most of you will probably not be doing algorithm development, but if you were, you'd still need to start with a gold standard or something stable that you can compare it with. As an INCF sponsored event, it's mandatory for me to tell you a little bit of what neuroinformatics is. So only 3 slides and we'll be done. Okay, so I personally view neuroimaging analysis as a sub-branch of neuroinformatics. Neuroinformatics is a new field. I think The INCF was formed in 2006, 2007 from an OECD, so it's actually European-centric in its inception, but has since grown to be a worldwide network. And the problem that INCF is trying to solve is, in layman's terms, I have big data for brain sciences. How am I going to use that? But that's an overly simplistic way of viewing it, and I just like to explain it from this tripartite model with this base here. Today, people are acquiring lots of data, tons and tons and tons of data. And it's not just data from one type of machine, it's data from many types of machines and data from all over the world. So it's easy now to actually get large databases and something called a knowledge base. And you can think about knowledge base as a summary, a conceptual summary of the raw data. Raw data could be something like numbers, observations, measurements. A knowledge base could be this set of numbers actually is correlated with this disease. This set of numbers actually indicates the presence of this disease. I have this observation, this phenotype. It probably means this for this patient. That's a knowledge base. And within the INCF network, there are people who are working on extracting these concepts from textbooks, from papers, and they're using machine learning for that. So you have all these acquired data and conceptual interpretation of that. On the other hand, you have people who are writing extremely powerful simulation tools, simulation models. There are various levels of models of the brain, like all the way down to the neuronal, the genomic models, the system, the circuits models, and how all of these interact. And if you're looking for a fine example, look at the Human Brain Project in Europe, where they've actually done a lot of computational simulations. And the idea really is that if you're just drawing interpretations from the raw data, that gets you so far, but perhaps not much more, because you don't have, at some point you run out of ways to explain the data, and you can't quite tell if you're just making it up or it's actually a real model. So when you start looking at the computational models, you have physical principles, you have neurological principles, you have principles that make sense, like the nerves are actually trying to reduce energy, things like that, to explain away this. And then you try to bridge the computational simulations with your real data and see how they fit and how they can actually help you to make the conclusions you want to draw. So I view neuroinformatics as the integration of data and knowledge bases with the computational models in order to create the sufficiently powerful analytical tools for you to answer your complex problem. And this is my definition for 50 years down the road. It's just that it's such a large and difficult problem because A, there's so many types of data. B, we have a lot of data, but not nearly enough statistical power compared to the diversity. C, our systems don't really talk to each other. So how can you share data? The data is not actually acquired in the same format. They're not actually acquired in a way that makes it easy for you to compare and integrate that. And so INCF and the neuroinformatics community, it's now actually focused on one major problem. And that's the problem of sharing our resources. What are our resources? Our data, our knowledge bases, our computational models, and the software and hardware tools that actually work on that. And to achieve this sharing, this is done a couple of ways. The first way is trying to get groups of researchers to agree, standardization. So the Docker, the NiPipe workflows, these are efforts in the standardization because they're amazing tools, AFNI, FSL, FreeSurfer, SPM, which were developed standalone, which not quite fit together. It's like scratching your head trying to actually fit output, intermediate outputs from one to another. And if every researcher were to do that, then we'd spend half or three-quarters of our time trying to fix the two problems. So one of the ways would be to standardize the way that software systems interact, and the bits format. Remember the directory structure where we actually went through. There's one directory for DWI, for functional, and for anatomical. So trying to get people all over the world to agree on a common exchange of data. Second, to integrate. That would be the workflows, trying to actually allow all these tools to work together. And third, training. Things like this course. to make you aware of these tools and get you started on the path to using them. So neuroinformatics is driven by this idea that there is a complex and important problem. You wouldn't actually use big data on a simple problem. I mean, if you can solve it with simple statistics, why go big data, right? It's silly. The problem is complex, and what is it? You're trying to improve health or performance, mental health or performance. You're trying to undo disease and degeneration. And I would say the brain is actually central to human science, right? Because no brain, no logic, no science. So it's a complex problem, and I hope you agree with me, it's an important problem. And there's data and observations that have to be made at multiple scales, and always think the scales are on two dimensions, the time dimension and the spatial dimension. So the time dimension would be rapid processors on the order of femtoseconds, maybe not femtoseconds, maybe on microseconds, milliseconds. This could be maybe protein expression, task, molecular exchange. Then on the millisecond range, it would be electrical connections between neurons, and on the seconds range, it would be chemical interactions between the neurons, and then on the days, weeks, months, could be behavioral interactions. So that's the time scale. And the space scale, you have whole brain regions, within nodes, within cells, within genome. So at all these levels, they are providing different sources of information. The question is, how do you integrate all of that to be able to solve this? Okay? And what's also interesting is it's a multimodal problem. All these data at all these different scales is obtained from a variety of different instruments, which have a variety of different interpretations. I mean, we just saw from MRI that there are at least two different interpretations of the T2 weighting. The T1 weighting means something different. There's MRS, and that's just MR, right? We've not talked about EG, FNIAS, MRS. We've not even talked about the animal model work, which could include genomics, genetics work, the optical and fluorescence microscopy, which are mainstay in a lot of labs, and all forms of microscopy, electrophysiology. Yeah. So it's trying to use and solve this multi-omic neuroscience data to solve a big problem. Okay, so that's my mandatory 3-slide explanation of neuroinformatics.